So in ETS, complex 1 is NADH dehydrogenase, in which electron enters, then FMN gets converted into FES. So you can remember FMN get converted into FES. Okay, next ubiquinone receives, or then you can see the ubiquinone ubiquinone receives the reducing equivalents from complex 2 and also complex 1. Next we have complex 3, which is cytochrome BC1, in which cytochrome B gets converted into FES, then it gets converted into cytochrome C1. Cytochrome C is a small protein which is attached to the cytochrome C is attached to the outer surface of the inner membrane of mitochondria. And next, cytochrome C is the mobile carrier between complex 3 and 4. Complex 2 here it is given, which is succinate dehydrogenase. It converts succinate to fumarate, and FAD is converted to FES. Then we have complex 4. Cytochrome 4 is the cytochrome C oxidase, in which copper A is present, then cytochrome A, then cytochrome A3, then copper B. So two copper complexes are present in complex 4. And next we have the last, which is complex 4, which is ATP synthase, in which F1 and F0 particles are there. F0 particle, so CF1 is the head piece, and it is the peripheral membrane protein complex, and it is site for ATP synthesis from ADP and inorganic phosphate. CF0 is the integral membrane protein complex, and it forms the channel through which the proton cross the inner membrane. Then the passage of proton through the channel is coupled to the catalytic site of component for production of ATP, and 4H plus passes through F0 from the intermembrane space then to matrix. Then we have glycolysis, which is a 10-step process. So we have first is the glucose get converted into glucose 6 phosphate, then glucose 6 phosphate converts to fructose 6 phosphate, then fructose 6 phosphate converts to fructose 1,6 bisphosphate. It gets converted into triose phosphate. Two forms of triose phosphate are like glyceraldehyde phosphate GAP and DHAP, which is dihydroxyacetone phosphate. Both are 3 carbons. Then it gets converted into triose bisphosphate, which is 1,3 bisphosphoglyceric acid. It gets converted into triose phosphate, which is 3 phosphoglyceric acid. 3 phosphoglyceric acid get converted into 2 phosphoglycerate. Then it's get converted into 2 PEP. Then 2 PEP gets converted into pyruvic acid. So the steps for ATP utilization are glucose to glucose 6 phosphate and fructose 6 phosphate to fructose 1,6 bisphosphate. The site for ATP formation are 1,3 bisphosphoglyceric acid to 3 phosphoglyceric acid, and 3 PEP to pyruvic acid. And next step for formation of ATP is from PEP to pyruvic acid. Then the step at which NAD gets reduced is from glyceraldehyde 3 phosphate. Okay, it is from triose phosphate to triose bisphosphate, which is at 2 forms, to 1,3 bisphosphoglyceric acid. and the step at which water is released are is from 2 phosphoglycerate to 2-phosphoenol pyruvate, and then uh, the net ATP synthesis in this process. To calculate it, we can see that the two ATP molecules are utilized, and here 2 plus 2, 4 ATP molecules are formed, right? And NADH plus HH plus is formed, which releases two NADH plus are formed, which releases like 3 ATPs. So 3 2s are 6, 6, this 4 ATPs, so 10 ATPs, and 2 are utilized above. So net ATP synthesis is like 8, and only how much ATP is synthesized if this is asked, then answer is 4, and 2 ATP, and net and total how much ATPs are utilized, answer goes 4, 2 2, sorry. Next, bryophytes. The first diagram is given of liverwort, which is mercantia. A is the female thallus and B is the male thallus. In female thallus, we can see the archegoniophore, then gamma cups are present, and then rhizoids are present. You can look at the difference in archegoniophore and anthridiophore in male and female. Next is the anthridiophore in the female thallus, also male thallus. We can see the gamma cups and rhizoids here. Next we have the mosses. Mosses example is funaria. So first diagram is given of funaria. It you can see the gametophyte and the sporophyte. So gametophyte here structure is rhizoid. Here you can see the rhizoid and main axis, then leaves, then seta, and then capsule. Next we have sphagnum, which is also moss, and in sphagnum it is the gametophyte. Sphagnum diagram is given of gametophyte. You can see the antheridial branch above, and then below is the archegonial branch, and branches are also given here. Next we have pteridophytes. First diagram is of Selaginella. In Selaginella we can see the leaves, then stem, and then roots. Next is the equisetum diagram. Here you can see equisetum. It has strobilus, then we can see the nodes, then internode, then branches, then rhizome is given. Next we can see the fern diagram. Next is the salvinia, right? So these are all the pteridophytes. Revising once again, pteridophytes examples are Selaginella, equisetum, fern, and then salvinia. Next we have cockroach. So elementary canal of cockroach. First is the pharynx, then we can see the salivary glands, then salivary reservoir, then we have esophagus, then crop, which is a sac like shape structure, then for grinding we have gizzard or proventriculus, then hepatic cae are present, then we have midgut or mesenteron, then malpighian tubules 100 to 150, then we have ileum, then we have colon, and then we have rectum. Next we have external features of cockroach, in which we can see the filiform antennae are present, then we have head, then compound eyes are present, then we have pronotum, then we have mesothorax, and first wing is called tegmina, second wing is called hind wing, and second wing, that hind wing is used for flying. Next, tegmina is a mosaic and dark colored wing. First segment was pronotum, then we have mesothorax, then we have metathorax, and the first pair of leg arises from the so yes, first pair of leg is the prothoracic leg, then we have mesothoracic leg, and then we have metathoracic leg. Tegmina arises from the mesothorax and hind wing arises from the metathorax. In diagram it is incorrectly given. Then we can see the abdomen, then we have anal cerci, and yes, next, then the frog structure is given here. You can see the head, then eyes are bulged out covered by nictitating membrane. Then we can see the trunk, then we have forelimb, hind limb, tail is absent and neck is also absent in frog. Next we have the skull, human skull diagram. So we can see here the frontal bone, then we have backside. On the backside we have parietal bone, then temporal bone is present, then occipital bone is present, then occipital condyle is present. We have one hyoid bone, then on the lower side we have mandible, on the upper side we have maxilla. In nose there is nasal bone, and we have then zygomatic bone, lacrimal bone. That small structure is the lacrimal bone. Behind lacrimal bone is the ethmoid bone, and behind ethmoid bone is the sphenoid bone. So learn like sequence wise: lacrimal, ethmoid, sphenoid, lacrimal, ethmoid, sphenoid, and in diagram also it is given in that order only. So nasal, lacrimal, ethmoid, sphenoid. Next we have female reproductive system of cockroach, in which we can see the oviduct is present, then ovary is present, then ova are present inside the ovary, then we have the ureter, then we have cloaca, and we have the cloacal aperture, then urinary bladder is also present. This is the female reproductive system, and oviducts are present above. Okay, next we have, and there is no functional connection with the kidneys in female reproductive system, and they open separately. Next we have the human respiratory system diagram of lungs, in which starting with epiglottis, this is a flap like structure which gets covered during the entry of food. Then we have larynx, then trachea, then we have the bronchus and this cut end of ribs. Then we have lungs, pleural membranes are present, which has pleural fluid which reduces the friction between the layer. Next, diaphragm is present on the lower side. Look at the alveoli and bronchioles carefully. Alveoli are given like a circular structure, and which divides into bronchioles. Then we have the pleural fluid is present between the pleural membrane. Next we have the human urinary system. This adrenal gland are present. Then we have adrenal glands are present above the kidney. We have inferior vena cava on the left side. On the right side we have dorsal aorta. So remember which is on the left and which is on the right. On the right side we have dorsal aorta, left side we have inferior vena cava, and we have then urinary bladder which opens into the urethra, and ureter opens into the urinary bladder. Ureter, kidney is shown here. Then we can see the small renal vein is shown. Then renal artery is present. The important point here is that the renal artery contains the highest concentration of excretory waste, and the renal vein contains the least amount of excretory waste. On the left side we can see the uh, inside the structure of kidney, we can see the pelvis is present, then medulla is present on the inner side, outer side cortex is present. Next, longitudinal section of kidney, we have the medullary pyramids, we have calyx, then we have renal columns. First we will look at the renal artery and renal vein, it was shown earlier also. So renal artery and renal vein, then we have the renal pelvis, then ureter which opens into the urinary bladder. Middle portion we can see it is the renal pelvis. Medullary pyramids are shown here, then renal column, then we have the cortex outermost, and renal capsule is seen here. Next, we have the male reproductive system of cockroach.